Hi, I'm Michael Breakspear. I'm at the University of Newcastle on Awabakal and Worimi country, and I'm presenting in the Wednesday morning Symposium, a talk entitled The Wave Particle Theory of Brain Function. This talk is in 4 parts. I'll first speak to neural mass models, then neural field models. I'll give a case study of sleep transitions in baby brains, and then present a brief overview of the thesis. This talk is based on computational modelling of large-scale brain systems, at the heart of which sits a so-called evolution equation. This is typically a differential equation, dx to t, where x is a vector that comprises a set of neuronal states. These could be firing rates, membrane potential, channel capacitance. These are not the observables, EEG, fMRI, MEG, but rather the underlying state equations. The evolution equation incorporates local circuit processes in, for example, canonical microcircuits for the cortex, inputs and outputs conveyed through the structural connectome. The states comprise to local average values. These aren't individual neurons, but local mean field values. How do we get there from complex underlying neural histology? Well, this can proceed in a number of steps, from taking the histology and reducing it to a complex multi-compartment spiking model with lots of physiological detail in each component of the neuron, such as embodied in the Human Brain Project. A simple spiking neural circuit model, where each point, if you like, is a single neuron. Brine, neuron, and spike net embody this. Or a lumped neural mass model, where each state is a local mean potential. And the virtual brain and the brain dynamics toolbox embody this, a level of description. Process of going from detailed underlying neural histology to increasingly coarse-grained descriptions is mathematically known as a mean field reduction. And you can also think of influences going the other way, where large-scale fluctuations capture and enslave smaller system components of the system. This is an old notion of the enslaving principle by Herman Harkin. But I'm going to be concentrating on neural mass and neural field models, where there are lumped or mean field population models, following the work of Walter Freeman, Paul Nunez, and others. In this particular demonstration, we'll look at neural masses exhibiting chaotic oscillations in each of our units, and we'll place these individual neural masses across the cortex and couple them together with data from the human connectome. We can put these into one of these large-scale simulation platforms and see what emerges. Now, in recent years, our group and many other groups have taken this approach and observed a variety of different patterns. Here we see rotating waves, breathers, which are outward-going waves, and cross-going traveling waves. Waves have been observed in a broad spectrum of functional neuroscience data, fMRI data, electrophysiological data, voltage-sensitive dyes across species, functions, cortical regions, and data modalities. The key thing here is an underlying local synaptic kernel whereby the strength between neighboring nodes in this model drops off exponentially as a function of distance. And there are some underlying long-range connections. Now we can go a step further with these neural waves, and we can actually pose our generative model as a damped wave equation, which I show on the top here. This model, developed by Peter Robinson, also includes corticothalamic loops and different populations in both cortex and thalamus. One can generate or predict through transfer functions healthy electrophysiological activity and pathological activity. Here we see seizure activity on the left, empirical, and on the right from the model. This is some old work I did in 2006. However, if you want to look at spatial activity, you can focus on what's called the Helmholtz equation, which is the spatial derivative, and you can solve the eigenvalue solution for this. And this generates a series of basis functions in the Laplace-Beltrami operator called eigenmodes. You can also look at a combination of temporal activity and spatial modes. This is an example we published some years ago, looking at flickering activity, alpha activity from an occipital mode. And this is a form of bistable fluctuations, supported on a single underlying eigenmode. So one has a decomposition into modes and the oscillations that they support, plus noise. This is the first 9 eigenmodes from this Laplace-Beltrami operator. And as you go to higher spatial frequencies, you also go to higher temporal frequencies. So now I'm going to present an example of this applied to EEG data acquired from newborn neonates and compare extremely preterm infants at full term with full term in neonates at term equivalent age. This data was acquired in Finland. We then proceeded to do source reconstruction and focus on the amplitude envelopes, and in this way, through linear correlations, reconstruct underlying networks, and then move on to explain them with neural field modeling. So if we look across both baby groups, across different sleep stages, we see a contrast using an ANOVA between active and quiet sleep, which are the precursors to REM and non-REM sleep. Quiet sleep is associated with this large blue network encompassing most brain regions, whereas active sleep comprises a smaller occipital region of predominantly short-range connections. We can then do a contrast of group and see this distributed network of nodes that are more correlated in the early psychosis group. The preterm group. And finally, with a 2x2 ANOVA, we can look at the interactions between sleep's transitions and preterm versus healthy term neonates. And in this occasion, we see that the transition from active to quiet sleep, or pink to blue, is attenuated in the early preterm infants. And this lack of transition preempts visual performance at 2 years of age. Now back to the sleep modes, we then sought a generative model of these observations. We took the model I showed before, we put a carrier alpha frequency with some slow frequency fluctuations, solved the eigenmode solution on the cortex, and generated the modes. Now, because our network contrasts were predominantly symmetrical and front to back, we just chose 3 spatial modes, modes 1, 3, and 4. We're then going to try and model the data on the left as a combination of the eigenmodes and the carrier frequencies plus noise. There's the carrier frequency and the noise, and then we try and get the best match for the modeled and empirical functional connectivity. We can do this for each neonate. It works particularly well. And we can re-express the contrast between groups and sleep stages now, not in terms of networks, but in terms of spatial modes. And we see in particular a group effect in the first mode, the global mode, which is attenuated in early psychosis, and no difference in the higher order modes. So concluding my brief overview of this complex area, this is what I mean by wave particle theory. Particles are waves being eigenmodes, and particles being nodes connected by functional connectivity edges. We can see that on the left, the neural field eigenmode description, and on the right, the neural mass and particle description. These are just two complementary ways of trying to describe and model generative processes in the brain. Thank you very much for listening to my talk, and I hope you'll be able to come and see this talk in person at Human Brain Mapping. Thank you.